Hello, welcome back. This is Professor Young, and this is the beginning of the end for our look into AMP2. Our final topic that we need to really kind of look at and discuss here is human reproductive biology, and we're going to divide this up into two key groups. This first session, which will actually be three different recorded lectures, is going to have us looking at male reproductive as well as a little bit about fetal development. And then the next set, which will probably be three separate sessions as well, we'll look at female reproductive as well as the menstrual cycle. And so for these next three sessions that we're going to be looking at, we're going to be specifically focused on male reproductive as well as some fetal development and what can go wrong. It's always fun when we discuss the abnormal stuff. And so that's exactly what we're going to be looking at. And so let's go ahead and get ourselves started here as we move through. It always helps to know what exactly it is that we are talking about. And so why don't we start there? When we talk about reproductive biology, the big picture, and the big picture, of course, is going to include male and female reproduction. There's a lot of things that we need to consider. We need to consider hormone interplay here. What's the role of the hormones on male and female reproductive systems? And really, we could stop right there because everything that you see listed underneath of this, puberty, menstrual cycle, pregnancy, and even sexual sexual identification or sexual identity disorders, all of these things, those last four bullets there, all specifically relate back to hormones. Puberty is directly linked and controlled through hormone regulation. Menstrual cycle, as we're going to find out, is directly linked to hormones. You know, you have luteinizing hormone, you have follicle stimulating hormone, you've got estrogen, you've got progesterone, you got oxytocin. What are all of these hormones doing? And how does that relate to that whole menstrual cycle? I mean, pregnancy, for heaven's sakes, that's definitely hormonal. You know, postpartum depression, what causes that? After the infant is born, you've got a major clash now of hormones that go on over the next 2 to 3 weeks, 4 weeks. And so what are the roles of hormones there? And as we are going to find out, you know, when you start looking at sexual identity disorders, all right, whether it is a hermaphroditic sexual identity disorder, whether it is brain development, masculinization or feminization of the brain, whether it's a genetic basis, whatever it is. For me, sexual identity disorders are any kind of a disorder where you've got a person that is questioning their sexual orientation, right, or their sexual being, who they are on the inside. And how does that translate to who they are on the outside? Everything that we've looked at right here all goes back to hormones. Really, reproduction or the reproductive systems is an extension of the endocrine system. And so we're going to be spending a lot of time over the next 5, 6 LIMBA sessions really looking at how these hormones play a role. But before we get there, we always have to talk a little bit about anatomy and structure. Here's the nice thing. Male reproductive system is a little bit more complex. But the hormone interplay is much simpler. When we look at female reproductive system, the organization of the female reproductive system is rather simple. But the hormonal interplay is a little bit more complicated. And so we kind of take a opposite approach with both of these. When we look at the male reproductive system, it's fairly straightforward. We're going to talk about a little bit more in detail the structure of the testes, but we know that we've got the scrotum, which is really the external genitalia compartment, if you would. It's where the testes are housed. It's within the testes that, of course, we have sperm production. This is where the sperm are initially begin to develop and produce. We'll talk about that. Once they are, as I like to say, once they go from being infants to teenagers, the sperm are going to leave and enter into the structure known as the epididymis, right there. The epididymis is kind of a playground for teenage sperm. It's where they learn to swim. It's where they continue to mature. Once they are mature little sperms, they will leave through the vas deferens. You'll see here that it's also referred to ductus deferens. I almost always exclusively use the term vas over ductus. And so on my assessments, you're always going to see that term vas deferens instead of ductus deferens. But you have vas deferens. The vas deferens is what is going to really link the scrotum, if you would, the epididymis and the testes, into the abdominal cavity. Right? Male reproductive, external male reproductive, presents a very unique challenge because you've got these, you've got the external genitalia that are literally external. They are outside of the rest of the body, but yet the product of that external genitalia, the product being the sperm, needs to go from outside of the body and pass back through the abdominal cavity, that inguinal canal, and enter back into the abdominal cavity. Now, the way it does that, the sperm are transported through these vas deferens, but the vas deferens is just part of the overall structure. The vas deferens are just part of the overall structure. Vas deferens actually are a part of a structure that we call the spermatic cord. And it's within the spermatic cord where we find not only the vas deferens, but we find nerves and we find blood vessels. And so you've got a rich supply of blood that enters down into the testes. You've got nerve innervations that enter down into the testes, as well as the vas deferens. And all three of them, the vas deferens, the nerve, and the blood vessels, the blood supply, are all encapsulated, if you would, within the spermatic cord. And then it's the spermatic cord that is going to provide the ductwork where it's going to pass into the inguinal canal. So, from the vas deferens, our little sperm are traveling up the spermatic cord through the vas deferens. It's going to enter into the abdominal cavity where it will empty into another paired structure, right there and right there. Notice that we are right below the urinary bladder, and that actually we're sitting behind the urinary bladder here. And so if anything, you're looking at this from a posterior view. Right? The vas deferens empty into a pair structure known as the seminal vesicles. Now, I like to describe the structure of the seminal vesicles as being almost like ram's heads. So if you look at a ram's head horn, look at the horns that come off of the ram's head, that's exactly what the seminal vesicles are going to look like. The job of the seminal vesicles is to add nutrients to the sperm. And so it's a high sugar secretion that the seminal vesicles are adding into the overall mixture. Keep in mind, we technically do not have semen at this point in time. You've got sperm, and now we've added nutrients. You got to feed those little boogers. It takes a lot of energy for those little tails to be whipping back and forth and ungulate their way through through the urethra and through the spermatic ducts, or the ejaculatory ducts. So from the seminal vesicles, nutrient is added. We pass down into the ejaculatory ducts, down into the next structure, which is the prostate gland. So the prostate gland sits below the urinary bladder as well as the seminal vesicles. The job of the prostate gland is to add fluid. And so we're adding in more of this component of semen. And so we have sperm, seminal vesicles. We've added in nutrient. Now we've added in some other fluids through the prostate gland. We continue down that ejaculatory duct until we get to a very small set of structures known as the bulbar urethra, or the other name that we go by for this. The other name of the bulbar urethra gland is Cowper's gland. Cowper's gland is, and that's just typically the name that I will refer to it as. Cowper's gland is there to add mucus into the semen. And so sperm travel up the vas deferens, passes through the seminal vesicles, which is going to add nutrient, passes through the prostate, which is going to add fluid, passes through the Cowper's gland, which is going to add a mucus secretion into that semen. The mucus is important because it will coat and protect the sperm through the initial entry into the vagina, which has a very acidic environment. You're talking about a pH of 3 to 4 within the vagina. And so those sperm need protecting, or that acidity of that area would go ahead and kill them off. And it does anyway. About 75 to 80% of the sperm don't make it out of the vagina itself because of the acidity. But that 30% or so that is surviving survives in part because of the protective mucus that is secreted through the Cowper's gland. Once you go through the Cowper's gland, then you enter into the urethra, and that semen is then sent down the penis and through the glans penis, which is the head of the penis, and is ejaculated out of the body. And so that's an overview of the male reproductive system. A lot of this stuff here I've already talked about, and so in true fashion, I'm not going to spend a whole lot of time talking about this, but you can see some of the key terms here. Spermatic cord again. We know that spermatic cord is again going to contain some very key structures. It's going to contain the vas deferens. It's going to contain the testicular nerve, and it's going to contain the blood vessels. And so that's kind of the key idea here that you need to understand with the spermatic cord. From the testes to the epididymis into the vas deferens. The vas deferens is encapsulated within inside of the spermatic cord, sends the vas deferens through the inguinal canal, passes externally to internally within the abdominal cavity. From there, sperm is sent into the seminal vesicles, nutrients are added, sent down the ejaculatory duct into the prostate gland where fluids are added, sent through the Cowper's gland, which is where mucus is added, down into the urethra, through the penis, passes through the glans penis, and then ejaculated right on out of the body. So not too much new, interesting stuff that is going there. You can see on this picture, since this is a side view or a lateral view, right here, that area that I have highlighted, that would be the inguinal canal. And so that is where the spermatic cord is going to pass from outside of the body, which would be right here, to within the abdominal cavity. And if you're really looking and interested, you're looking at, let's see here, what I'm trying to do. This bone, this structure that you're looking at right here, that I've just highlighted, that would be the pubis bone of the hip. And so just kind of give you an orientation as far as what you're looking at there. So that's what you're looking at. Now, within the testes, the key process that is happening throughout this stage of development is known as spermatogenesis. Spermatogenesis is the production of sperm. Spermatogenesis is the production of sperm. And sperm production, spermatogenesis, specifically happens within these tiny tube-like structures that are found within the testes, known as the seminiferous tubules. The best way to explain it is ramen noodles. They look just like ramen noodles or oodles of noodles. And for college students who probably are not making a lot of money and who are either living at home or living on campus, you are probably very much so familiar with ramen noodles because that's probably a staple of your diet. And I've now just ruined eating ramen noodles forever and ever for you. Because now whenever you see ramen noodles, you are going to think of sperm production and seminiferous tubules within the testes. And so you can thank me later for that image. I hope you're not eating ramen noodles right now as you are listening to that, because that would be kind of gross. Within the seminiferous tubules, there are two main types of cells. You have germ cells, Spermatogonia, as they are also referred to as, but you do have germ cells. Germ cells or Spermatogonia is where the sperm are initially going to develop. Once, and really what's happening with the germ cells is mitosis. These germ cells are going to divide. They're going to go through mitosis. They're going to have a full diploid count as far as their chromosomes go. And once they reach a certain level of development through mitosis, they will then go from the germ cells to the more superficial layer of cells, which are known as the Sertoli cells. Now, Sertoli cells are all about feeding. Right? They provide nutrients to the very immature sperm. But the Sertoli cells allow the sperm to go from being juveniles to being teenagers. And once those sperm have reached that quote-unquote teenage setting, that is when they are released from the Sertoli cells and then can leave the testes and enter into the epididymis. Now, how they do that, we'll talk about in a little bit. But know that that happens for right now. As I've already mentioned, Spermatogonium is the name that we refer to. Oh, that was dumb. Is the name that we can also refer to the germ cells for. And again, I cannot stress here the fact that this here is a process of mitosis. And what we mean by that is we have a period of cell division with full diploid chromosome count. We do not have the process of meiosis going on here. This is strictly mitosis that is happening within the germ cell. The other thing is, notice the hormone that is responsible for the stimulation of mitosis within the germ cell. Estradiol, which is a derivative of estrogen. Actually, estradiol is a precursor to estrogen. Estradiol can be cleaved and changed into estrogen. Where's the estradiol coming from? Estradiol, ironically, is coming from testosterone. Testosterone has the ability to be transformed into a number of different hormones, as you will learn. One of those hormones that testosterone can be cleaved into is estradiol. Now, here's why this is critical. Here's why sperm, and this is kind of ironic. Think about this. Sperm is under the direction of a female hormone derivative. Not a male hormone. And the reason for that is because the germ cells lack testosterone, sorry about that, receptors. The germ cells do not respond to testosterone. There are no androgen receptors. There's a term for you that you need to become familiar with. Androgens are basically any of your male hormones. Testosterone, DHT, DHEA, anything like that. Anything that's a derivative of testosterone that's not a female hormone is considered an androgen. It's a male hormone specifically. And so germ cells, Spermatogonium, lack androgen receptors. They lack the receptors to allow testosterone, and specifically DHT, dihydrogen testosterone, dihydrotestosterone, to bind. And so the spark for mitosis within the germ cells is estradiol, which is a derivative of testosterone. Make sure you understand that. Make sure you understand that when we talk about Spermatogonium undergoing mitosis, that means we are increasing the number of cells, but we are not reducing the chromosome count. It is still a full diploid number. Don't lose sight of that. If you lose sight of that, you're going to be dead in the water. Now, this is where we begin to get into meiosis. Keep in mind, meiosis has two separate processes. Meiosis 1, which is all about genetic recombination. It's during prophase 1 of meiosis 1 where we get things like tetrads. And once the tetrads are formed, crossing over occurs. And then once that we've shuffled that genetic mix, meiosis 1 produces two new cells that are haploid in chromosome count. And so our primary spermatocytes that were just formed from germ cells through mitosis now enter into meiosis 1, where we shuffle the genetic makeup of it, and we go through the process of meiosis 1. At the end of meiosis 1, we have two new spermatocytes that are haploid. And remember, when we talk about something being haploid, what we're saying is, specifically humans, we've taken that chromosome count of 46 chromosomes and we've reduced it to 23. So now each of our new little spermatocytes that have been produced at the end of meiosis 1, instead of having 2 copies of each chromosome equaling 46, they now have 1 copy of each chromosome. 1 full copy of each chromosome. Then meiosis 1 leads into meiosis 2. Meiosis 2 has the distinction, if you would, or the responsibility to take that haploid chromosome count and now, still being haploid, split the chromosomes so that each new spermatocyte, each new little sperm that is produced, now has 1 chromatid from each chromosome. Each of our little spermatids at the end of meiosis 2 has 1 copy or 1 sister chromatid from each chromosome. Each new spermatid has 1 copy, 1 sister chromatid from each chromosome. Remember, here's my very crude chromosome. That right there is a sister chromatid. This right here is a sister chromatid. And so each sperm that is produced by the end of meiosis 2, which, by the way, remember we end up with 4 sperm at the end of meiosis 2. Each one of those sperm receives 1 chromatid of each of those 23 chromosomes. Makes sense. Try to digest that. If you need to stop this session, rewind it, and listen to it again, do it now. Do not go any further if you do not understand what was just discussed. And after you listen to it 14 and a half times, if you still don't understand what I just explained here, make sure you shoot me that email. Don't allow yourself to go any further until you understand exactly what we just discussed here. Now, I will say this. What we just discussed is exactly what is on this page here. What we just discussed is exactly what is on this page right here. Here is the seminiferous. Here's a cross-section of a seminiferous tubule right there. This region down here would be the lumen. It's the middle of the seminiferous tubule. The green box there is the seminiferous tubule. which means this region right up here, and I will put a nice little line through this. This upper portion of the seminiferous tubule is where the germ cells are found. That is where mitosis is happening. Then we cross that line, we cross that imaginary line, and we enter down into where the Sertoli cells are. And it's where that second region, it's that lower part of the seminiferous tubule where we've got meiosis 1 and meiosis 2 happening. And so we take the cells that we have produced from the germ cells through mitosis. We've increased the number of cells without changing the genetics. They're still diploid. These guys here are all diploid. Once they enter down into this region, this lower region, then they become haploid. And they become haploid because of meiosis 1 and meiosis 2. So take what we just looked at on the previous page and apply it to this visual representation of now what you are looking at. And it should make sense. It should. That's the hope. Remember, I can't stress enough, if you're not sure of what we just talked about, go through and rewind this. Just rewind it. All right. Spermatogenesis. We talked about spermatogenesis being the process of sperm production. It starts from the germ cell, and it goes through final maturity within the epididymis. That's the whole process. Because it's not until the sperm enter into the epididymis that they understand fully how to swim. In other words, the Sertoli cells, which are really, it's almost like a nursing cell, because the sperm inserts its head into the Sertoli cell, and they gain nutrients from that. At that point in time, once they're nursing off of the Sertoli cells, they become teenagers. Well, think back to when you were teenagers. You did a lot of stupid things, probably. Sperm do the same thing when they're teenagers. They do a lot of stupid things, like they swim in circles. And so they go to the epididymis to learn how to swim straight, like a good little sperm. And so spermatogenesis needs to be able to continue through the process of going through the epididymis. The Sertoli cells are there within the seminiferous tubules to provide nutrients and development of our new fledgling sperm. And here is what it looks like. And so if we were to kind of separate this all out, that is not going to work the way that I wanted it to. Hold on 1 second. And that is not going to work the way that I had hoped. Are you serious? Aha! This is going to work. So if you look at my crude division here, your germ cells are up in this outer area. Your Sertoli cells are located down here in that inner area. The lumen is right here in this area. And you've got your little old sperm with their heads in their lumens, or with their heads in their Sertoli cells, all right there. That stringy stuff that you see going into the lumen are the tails of the sperm. So that's what it looks like underneath the microscope. Give you a little bit closer up view. So here specifically what you are looking at are this region all right in here are all Sertoli cells. And you can see the sperm and their little old tails right there. All right? Pretty simple, pretty straightforward. Again, here, this is just some basic information. I don't think I need to spend much time on this. What is important here is that Sertoli cells have the ability to convert testosterone into DHT. Now remember, DHT stands for dihydrotestosterone. And that's what DHT stands for. And keep in mind that dihydrotestosterone is just a amped up or souped up version of testosterone. It's a more potent version of testosterone. But again, there's nothing here that you can't read and I think understand on your own. This is fairly straightforward. And again, I made mention to this earlier. We know that sperm eventually will leave the testis and enter into the epididymis. How they do that is outlined right here on this page. Right? And so from the seminiferous tubules, sperm are released, and they're going to enter into a structure that is referred to as the tubuli recti, then into the rete testes, and then finally into the vasa efferentia. The vasa efferentia is what is going to connect, if you would, the testis with the epididymis. That's the final branch. And again, on this slide right here, we've got a nice little picture of just that. So if we come over here, hold on one second. So if we come right on over into here, we can see, I'm going to color in this whole area right in here that I'm outlining in blue is all seminiferous tubules. So that's all seminiferous tubules right there. The red line represents the tubuli recti, tubulus recti. Right, so from the seminiferous tubules, they enter into the tubuli recti, then into the rete testis, which I am going to put a box around as being right in. Oh, that didn't work. Hold on one second. I'm not going to put a box around it. I'm going to draw a box around it. How does that sound? Here's your rete testis. Right there. And then I will highlight for you once again in yellow. This here is the epididymis. Right here. The vasa efferentia, if you would, is, let's see, what am I going to do? What I'm going to highlight in green, and that's this region right there. So you have seminiferous tubules right here. You've got the tubulus recti right here. From the tubuli recti, you have the rete testis right there. From the rete testis, you've got the vasa efferentia. And then from the vasa efferentia, you enter into the epididymis. Right, so that's kind of the flow there. And then really these last two slides are simply a review. Right, we know that once the sperm travels through the epididymis, it starts at the top, comes down to the tail or the end, the bottom of the epididymis, which matches up with the bottom of the testis. That's where the vas deferens enter into. From the vas deferens, we curve up through the spermatic cord, passing through the inguinal canal. We attach to the back end of the bladder, the base of the bladder, which is where we connect into the seminal vesicles. From the seminal vesicles, of course, that is where the food source, the nutrients, is added into the sperm. Then we go through the ejaculatory duct into the prostate gland, which is where more fluid is added. From there, we go back down into the ejaculatory duct. We pass through the Cowper's gland, which is where the mucus is added into. From the mucus being added, we now fully have legitimate semen, and that is where we then enter back into the urethra. From the urethra, down through the penis, through the glans penis, and then out of the body. Right? And that's kind of where we are going to end this session of the WIMBA. Our next session, session 2, is going to look at hormone interplay here and what the hormones do. And then from there, and the roles that they play, and we're also going to talk a little bit about fetal development with this as well, and the role that the hormones play on fetal development. In other words, how do we go from this sexually indifferent character, this developing fetus, to being male or female? And then we'll end with when hormones go wild, and we'll look at some of the disorders that can happen through this process. And so with that said, I will leave you to your review and your study. And as always, if you have any questions, please do not hesitate to shoot me an email and ask me. Jot them down. Ask me at the beginning of the next class. Whatever you feel comfortable with, just make sure that you don't leave any questions unasked. Because that means that you're not understanding something, and that's my primary goal, is to make sure that you understand the material that we are looking at. And so on that note, happy studying, and I'll see you soon.